All right, welcome. This is going to be a little recording just to show you how to use some fairly common microbial genetics techniques to manipulate a microbial genome in your way of choosing. You can use these techniques to make a clean deletion of an ORF within a genome or any region within a genome. You can use them to do allelic replacement. You can even do them to put new genes into a genome and select for putting new genes into a genome in pretty much any place on the chromosome you would like to do. So first things first, I'm going to introduce you to the region of the genome that I'm going to be manipulating as part of this little module here. I'm going to work with YFG, which is your favorite gene. The first thing we have to do for actually manipulating the genome is to identify regions upstream and downstream of this YFG region that we actually want to manipulate. I've got the upstream region here labeled A. I've got the downstream region here labeled B. A is in blue, B is in green. It's important to identify these bracketing regions to the region you actually want to manipulate, because those are going to be the ones we use to actually homologously recombine the plasmid into the genome and take advantage of the ability of homologous recombination to kind of move around regions of DNA where we'd like them to be. All right, how large do these regions have to be? The larger the better. Long story short, the larger the better. It's homologous recombination we're dealing with, so the larger the region that we can do on either end of the region we want to manipulate, the easier it's going to be to actually select for integration of plasmids into the genome and to select for plasmids to flip out of the genome in hopefully the way we want. Right. Typically when I do this, when I build them within the genomes, I'm using about 500 base pairs on each side of the region I want to manipulate. I've gotten away with about 100 base pairs on each side. You can even go a little bit shorter than that. It just makes your life a little bit more difficult when you're trying to manipulate the genome. We typically go with 500 base pairs, but again, the larger the better. Thousands of base pairs would be even better than 500 base pairs here. All right, so we've got region A, upstream, region B, downstream. We'll say it's 500 base pairs each of those, and we'll kind of go from there. So what do we actually want to do to manipulate this genome? So I'm kind of fond of making clean deletions within genomes to actually knock out my ORF of interest or my region of interest. So what is a clean deletion? Clean deletion basically means you're going to take those two bracketing regions, A and B here, and just smush them together, cleanly deleting start codon to stop codon, the ORF of interest for YFG, or whatever region that you want to actually delete from the genome. So I call it a clean deletion. This is going to be a scarless deletion. It just it's as if the gene or ORF doesn't even exist within the genome. So that's what we're going to do. That's the goal of kind of what I'm going to show you these techniques today. Again, as I talk about at the end, you can use these techniques for a variety of other things as well. I'm going to introduce you to a plasmid I created, or Mark Nishimura created about a decade ago. We use this widely in pseudomonads. It works in some other things too, and we just use it really well to manipulate genomes of a lot of different gram-negative bacteria. This plasmid has a narrow host range, so it replicates well in E. coli, doesn't replicate in other things like Xanthomonas or Pseudomonas, and so it's useful as a plasmid with a conditional origin of replication where we can actually have it replicate in E. coli, have E. coli, manipulate it in E. coli, and then conjugate it over to other strains where we can actually then select for the plasmid to integrate into the genome. This plasmid's MTN 1907. It's got a few different features on it that I just want to point out here. There's a multiple cloning site. We've made a gateway compatible version, but originally it was a just regular old restriction enzyme cloning version that you could use. There is a gene for TET-A, which provides tetracycline resistance. There's also a dual cassette for NPT2 and SacB. NPT2 is kanamycin resistance. SacB is going to be for sucrose sensitivity. The kanamycin resistance on this plasmid is a little weird. The promoter doesn't necessarily work in pseudomonads, and so this plasmid actually provides kanamycin resistance in E. coli and tetracycline resistance in E. coli, but we can really only use it to select for tetracycline resistance in things like Pseudomonas. It's a promoter thing. Right, so that's the layout of the plasmid. Now let's dive into the SacB gene. All right, so the SacB gene was originally identified in Bacillus, a strain of Bacillus. SacB stands for Levana sucrase. It's a gene usually involved in an operon that manipulates sucrose and turns it into something else, as we'll talk about in a second. SacB is an enzyme that can take sucrose and this other long named thing over here on the left. Basically, manipulate them, change them around a little bit. It turns sucrose into something called leaven. Leaven, I'll just kind of click through here again. Leaven is actually toxic to many gram-negative species. And so what this gene does is it allows for counter selection, allows for negative selection against the presence of sucrose if you grow your gram-negative bacteria on a plate with about 10% sucrose. And it does this because the SacB gene will enzymatically convert sucrose into leaven. Leaven, we don't quite know how it actually kills the cells. There's a little bit of mystery about that. But it's really good at killing cells. It basically, we think it might get into the periplasm and kind of gunk up the works there. All right, so SacB is a good counter-selectable marker. So we've talked about positive selection before in terms of antibiotic resistance, where you can select for genes to go into a genome. Negative selection or counter-selection is something where we can select for genes to come back out of a genome. And so that's where the SacB becomes useful, because we can actually use it to select for things like the plasmid to flip back out of the genome, as I'll show you in a few slides. All right, so we'll run through the process from start to finish here, because it's a little bit, you kind of have to wrap your mind around some of the recombination steps here a little bit. All right, we're going to start off with our region of interest, again, A and B bracketing YFG. We're going to make a version of this where we're going to basically mock up in DNA a version of what we want the genome to look like. I want to make this a clean deletion, so we're going to build a synthetic piece of DNA where just A and B are smushed together without YFG there. You can do this through just ordering genes online now. You can do it through splicing overlap PCR. You can do it through some ligations. There's a few different ways to do this, but really it's just easiest to order these now online. All right, so we've got our synthetic piece of DNA made with the desired change of interest, in this case a clean deletion. What we're going to do is we're going to clone this into the multiple cloning site of 1907. So we clone that into the multiple cloning site of the 1907, and you'll just note I've changed the notation here on the A and B regions. I've just called them A prime and B prime to denote which region's coming from the plasmid and which region's actually in the chromosome. So just take note of that. All right, so we've got plasmid 1907. It's replicated in E. coli. There's an ORET on plasmid 1907, so it'll actually conjugate out of E. coli into a destination strain, but it's only going to replicate in E. coli. And so once it conjugates over to something like Pseudomonas or Xanthomonas, this plasmid's not going to replicate. So the only way to provide tetracycline resistance for this TET-A gene to actually function is to integrate into the genome and be replicated as part of the genome, and thereby allowing for tetracycline resistance, the TET-A gene to actually, the protein to actually be made. How can it actually integrate into the genome? Well, we've provided all of the homologous, the similar sequences it needs to actually do this A prime region and the B prime region. They're going to be sequence identical to regions on the genome where we actually want the plasmid to integrate into. So all we have to do is kind of get this plasmid into the strain of interest, and then let things like RecA do the work. RecA is going to find those similar sequences, comb through the chromosome, and then integrate this similar sequence into the chromosome. It's going to happen at a low frequency, but we're dealing with microbial genetics here, so even 1 in a million events we can select for pretty easily sometimes. All right, so what does this look like functionally? Well, we've got our plasmid, we've conjugated it into the strain of interest, and now it's integrated into the genome. I've shown this here by kind of denoting both the A and A prime and B prime regions as where they came from the plasmid itself. Again, the plasmid just smushes its way into the chromosome through recombination, and so it's going to be replicated as part of the chromosome. And because it has that tetracycline resistance gene, we can positively select for this plasmid to integrate into the genome by growing strains on tetracycline. This is a positive selection for the plasmid to integrate into the genome. You won't get tetracycline resistance other ways because the plasmid won't replicate in the destination strain of choice. All right, so going back to what it looks like in the genome here, we've got our plasmid 1907 replicated into the genome. A prime A, B prime B. You can kind of see where all the gene pieces come from. And so now all we're going to do, once it's in the genome, the strain is going to be tetracycline resistant, and it's going to be sensitive to sucrose because SacB is actually there. So all we have to do is take the strain, grow it up in non-selected media, so media that doesn't have tetracycline in it, and then just through the magic of RecA, there's going to be some recombination steps where the plasmid can then recombine out of the genome. It can recombine in two different ways, as I'll show you in a second. So really, we're just hoping it recombines in the way we want versus kind of recapitulating the wild type sequence. But really, it's a chance thing. It's going to happen one way versus the other, and we just have to get lucky and screen through for the right recombination event to isolate it. All right, so what does this recombination event look like? Well, if it recombines one way, it'll basically recombine sequences so that the wild type sequence, the wild type chromosomal sequence, is remade. And as this is basically the plasmid flips out through homologous recombination through the left A and the right A. We don't want these. This is remaking the wild type strain, so we want to avoid this, but it happens a lot, and you just kind of have to screen through and find the one of interest. How do we actually select for the ones that don't have the plasmid there? Again, we're going to grow this on 10% sucrose media. The sucrose is going to select against the presence of SacB, and so we're really just selecting for strains that have flipped the plasmid out, providing sucrose resistance in this case. And so that's recombination one way. We can also recombine things the other way, in the way of interest, where we actually get the clean deletion. And so in this case, again, it's going to happen in the same non-selective overnight culture as before, but we're just going to try to screen through and find the recombination event of interest. And what does this look like? Well, there's going to be a homologous recombination between the left B prime and the right B prime and the right B, leaving the only region in the genome looking like the clean deletion as we want it. So we've used recombination to flip in the plasmid with the deletion construct into the genome, and now we've used sucrose sensitivity to select against the plasmid to recombine out of the genome, and now we're screening through to find recombination events that happen this way versus the wild type way. And so really it's just kind of putting in the work to screen enough colonies to find the one of interest that's sucrose resistant and has the deletion like we want it. All right, when I showed this originally, I showed the plasmid integrated into the genome one way, but it can actually go into the genome a couple ways, which I'll show you in one slide. But first, I just want to highlight again that SacB is going to provide sucrose sensitivity, so it allows for counter selection against the presence of the plasmid. All right, like I mentioned before, the plasmid, when it integrates into the genome, can integrate in couple different ways. Before we showed it where it kind of broke up the A regions of the genome, the A brackets of the genome. Now I've just shown you an orientation here where the plasmid is integrated into the B regions of the genome. Same deal, it's just basically it can go in through recombination in any of the similar sequence tracks that we've provided for it with the A prime and the B prime sequences. But in this case, the plasmid's gone in and B prime is recombined with B, and it's just kind of flipped around the orientation of things. But this construct will work just fine for generating deletions too. We basically just want to select for the tetracycline resistance to get it into the cells, into the chromosome, and then we're just hoping to flip it out in the right way. All right, lastly, so that's the steps through to create a clean deletion. This is a pretty powerful technique though, so you can actually use it in a variety of other means to generate different changes in the genome as you would want. So we can take our wild type gene of interest, and we can actually just do allelic replacement the same way we do a clean deletion. We just basically synthesize our DNA so it has allelic replacement instead of a clean deletion, as I show on the bottom there. Likewise, we can use this construct as a landing place for completely non-homologous sequences within the genome, as long as we bracket it with those A and B regions that can actually then recombine to the genome. So we can use this technique and these plasmids to actually put new genes, novel DNA, whatever we want, into very specific places in the genome. And then it's just a question of screening through, finding the recombination event that we're looking for, and then you've manipulated the genome in the way of interest.